And what we have today for agronomic presentation, we have Sherrilyn Phelps. Sherrilyn manages the Pulse Agronomy program with Saskatchewan Pulse Growers. She has 25 years of experience in agriculture with expertise in pulse production and agronomy. She obtained her Master of Science from the University of Saskatchewan and has enjoyed her career filled with field research and extension. Her current role as Agronomy Manager allows her to develop programs best suited for advancing Saskatchewan's pulse growers through increasing yields, reducing risks, and adoption of new pulse crops such as faba beans and soybeans. She currently resides south of Battleford, Saskatchewan on the farm with her husband and son. So thank you, Sherrilyn, and I'll turn it over to you.
Thanks, Lori. So yeah, I was really pleased today to be able to talk to you guys a little bit about agronomics, kind of the hot topics with pulses and sort of variety, linking that to the variety implications as well. So what I'm going to kind of focus on is some of the hot topics that I've been hearing about during last summer as well into this fall and winter. Talking a little bit about seed quality. We do an annual survey each year, and we have the interim report now ready for what the results are that we're seeing with pulses. So I would like to share that information with you, as well as the chickpea field health issue, a little bit of an update on that. And then also talk a little bit about fungicide insensitivity, anthracnose and lentils. And of course, it wouldn't be a pulse talk unless I'm talking a little bit about root rots. So to start off with, talking about seed quality, we all know how important having seed tests and knowing what our germination and vigor is, as well as our 1,000 kernel weight. It also gives us an indication of what the disease levels are on seed. And like Kirsten mentioned earlier, that is, the disease levels can be critical, because if we don't know what they are, we can end up like the situation she mentioned and show the picture of the halo blight that was in the dry beans. And that can be very devastating. By knowing our 1,000 kernel weights, it also allows us to do the proper seeding rates. We can use the, average seed weight that's in the guide, but we know that that's different between varieties. And we also know that individual lots can vary dramatically. So today I'm going to focus on the seed quality report that is just hot off the press, and that's going to focus on the disease side of things and what we're seeing on seed for peas, lentils, and chickpeas this year. We have 4 labs that participated in the disease survey. However, London Seed Labs does not provide data till the very end, so I don't have the interim results from them. So this is data that's coming out of the Prairie Diagnostic Seed Lab, 2020 Seed Lab, as well as Discovery. So to start off with, I just want to talk a little bit about peas. So what I have here is a graph that's showing the different disease levels on seed from the past 6 years. So we can kind of compare our seed quality to previous years. So the first chart on the left is the percent pathogen-free samples. So that means the higher the number, the healthier the seed lot. So 100% means that there's no disease on the seed. We can see that with the blue and the purple, which is sclerotinia and botrytis, we have pretty good disease levels in terms of that there's a high percentage of pathogen-free samples. When we move down to ascochyta, which is usually the one that we're most concerned with peas, we can see that those numbers, especially in 2016, which were a bad disease year, have dropped a little bit, but are comparable to last year. When we look at the mean percent infection, so that is out of all the samples that were done, where there was positives for disease, what the average percent infection is. And we can see that with botrytis and sclerotinia, very low, but with ascochyta, we get that peak in 2016. Now this year coming out, we're showing about an average of a 3% level of infection of ascochyta on those samples. And just a reminder that our our levels or thresholds on seed for peas and ascochyta is at about 10%. And I'm going to break this down just a little bit more in terms of where the hot spots are. So we have just over 400 samples that were on this interim report. The highest disease levels were out of the Northwest. So when we're looking at sourcing seed, or when growers are looking at sourcing seed, we may see that there is more seed being bought in this Northwest area because there may be a higher level of disease on their own seed. So just something to keep in mind, although the numbers still aren't alarming, they're in that 4 to 6%, which is still usable as long as you're looking at using a seed treatment. When we look at lentils, so again, the pathogen-free samples, pretty good for the most part for all the diseases. We've added anthracnose in here, and we can see that the line starts to drop in 2020. We did see some anthracnose showing up in lentils this year in early July, and then it turned hot and dry, which tended to keep it somewhat under control, but we did see are seeing higher levels on seed. When we look at the mean percent infection, again, all the seed lots are fairly low in terms of the various diseases, but we are starting to see a little increase in the anthracnose just based on the interim numbers. When we look at this in terms of where in the province, the hot spots for the anthracnose is in the high lentil growing areas. These were the areas that had higher levels of, or lower levels of pathogen-free seed. When we look at the disease levels on seed, we saw that there was higher levels from these two crop districts. Still well within the safe levels on seed. With lentils, it's a 5% level for, 5% level for Ascochyta. With anthracnose, it's not really considered a high risk on seed, but if we're growing lentils and introducing seed with anthracnose into areas where you haven't had anthracnose issues, that is something to be cognizant about. When we look at chickpeas, Ascochyta is normally our biggest concern. Botrytis sclerotinia, fairly high in terms of pathogen-free samples. The Ascochyta, very similar to what it was last year in terms of seed quality. So even though we did see some Ascochyta out in the fields this year, the levels on seed have been relatively low compared to some previous years like 2015, 2016, and 2019. When we look at the area, again, we're looking at that chickpea growing area, the hot spot being that 3AS crop district. And that also coincides with the disease survey on Ascochyta, where the highest levels of disease were found in that same area. So not surprising that the seed coming out of there is showing a higher level than some of the other areas within the province. Now, the safe rate for Ascochyta on seed is the 0.3%. So it is slightly different. And this just kind of summarizes what the thresholds are. With peas for Ascochyta is 10%. Above that, use a seed treatment. Lentils, 5%. Above that, use a seed treatment. If it's above 10, do not use a seed. With chickpeas, that 0.3%. And then pulses in general, when we add up the botrytis, fusarium, and sclerotinia, if that number becomes 10%, then we want to for sure be looking at a seed treatment. A little update on the chickpea health issue. For those that maybe are a little more familiar or from that area, you're probably well versed in the situation that was going on. But for a lot of us, we're not down in that area on a regular basis. So I wanted to show what some of the symptoms were that growers were seeing. So in a lot of cases, it was starting as a bleaching along the outer surface of the leaves. And then it would lead to this leaflet blighting, where the tip of the plant or the top of the plant would start to die back, and individual branches, those new leaflets coming out, would also wilt and die back, and we would get premature leaf drop. In some cases, it was entire fields that were affected, and it was more that situation in 2019. But there was also a lot of cases where the issue was restricted to compacted areas, such as where the tractors, the combines, and the seeders would have gone, or just individual smaller patches within a field. To try and isolate what some of the causes of this issue is, we're looking at a number of different things, and Dr. Michelle Hubbard from Swift Current has been instrumental in looking at this health issue. And the one thing that we did notice that was consistent is that the majority of the area, or the majority of the infected fields, had dry conditions followed by a rain event in late June and early July. And here's a graph that she had developed from 2019 showing that Coronach, which was sort of the hot spot 2 years ago, did have fairly dry conditions in May, and then it rained, heavy rains at the end of June and into July, more so than other areas down there, and similar situation in 2020. What we've done in order to try and isolate the cause is we did do a bunch of plant samples from 16 different fields in late July this past year, and thanks to all the agronomists and growers who allowed us to take samples from their fields. We looked at herbicide residues, and nothing is showing up in there, and other than, you know, wherever residual herbicides were used, we are able to detect them. Nutrient deficiencies, nothing alarming there, but we are doing a little bit more work on the potash side and the electrical conductivity side. And then in terms of pathogens and viruses, there's nothing alarming that's showing up. Now, this is a really busy graph, but I just wanted to show you here that we did analyze for a number of root rot pathogens as well as a number of foliar pathogens. And the white bars are the healthy plant tissue, and the red is the unhealthy plant tissue or unhealthy plants. And we can see that there's really not a difference, suggesting that none of these diseases are really any of the one cause that's leading to the chickpea health issue. When we talk about varieties, now linking this health issue to varieties, originally in 2019, it was thought to be variety related, with CDC Orion more susceptible than CDC Leader. That caused a shift in the chickpea varieties this past year, and a lot of people switched from Orion back to Leader. In 2020, we found that the chickpea health issue showed up in both varieties, and not only just both varieties, pretty much every chickpea variety out there, as well as Kabulis and Desis. So it's no longer believed to be kind of variety specific. One of the things that we did realize was that the flowering time is slightly different between CDC Leader and CDC Orion, in that Orion flowers earlier than Leader, so it may have been just at a more susceptible stage earlier than Leader was, say in 2019, and that's why it might have been showing up in Orion in a little bit more prevalence. The next step, so what we've done since the summer field samples, is we've gone out to a number of fields and did soil samples. This time we found some fields that were relatively healthy, so we'll have some healthy fields to compare to the unhealthy fields. We're doing DNA scans for soil pathogens, as well as for the foliar pathogens. We've got the DNA from the lab where we sent for the plant tissue analysis. We're able to send us the DNA from those, and we can run both the soil and foliar scans, and that's being done at Agriculture and Agri-Food Canada at Saskatoon. We're doing some more further soil analysis, as I had mentioned, with the potassium and the electrical conductivity. We're also looking at nematodes at the University of Manitoba. There's also a new project that's starting with Sean Sharp, Michelle Hubbard, and Jeff Shano, where they're trying to recreate the symptoms, looking at herbicides, looking at stresses, looking at various different characteristics to try and control the environment so that we can see if we can simulate some of these unhealthy chickpea symptoms. So fingers crossed that we can kind of come up with a solution for this. My fingers are really crossed in the fact that maybe we won't have as much of an issue going into 2021, but time will tell. A little bit on fungicide insensitivity, so switching gears. I wanted to talk about fungicide insensitivity because this is becoming a hotter topic, and it's really, you can relate it to herbicide resistance in weeds. So this is insensitivity in, say, anthracnose. It's insensitivity in diseases to the application of fungicides. There has been work done in the past, so we know that we've had Ascochyta that has shown some resistance to group 11, group M5s, as well as group M3s. Just recently, we found group 11 resistance in anthracnose. There was a lot of samples that were taken at the end of 2019 that were really high in their level of resistance. And there's actually just been a survey completed in 2020, and the samples are sitting in the lab, so we don't have an idea of where our baseline is with the anthracnose. And then Microspirella in peas, we've also seen some strobilurin resistance or group 11. So we've actually developed a really in-depth fact sheet, and I'm going to go through this quite quickly, but you can definitely get more information out of the fact sheet. The big thing I wanted to focus on was choosing varieties with increased disease resistance as one of the main tools for integrated pest management and managing for reduced insensitivity development, as well as a tool to manage disease. The other factors that can help with the insensitivity management is good seed quality, rotation, scouting, as well as fungicides, only applying them when risk-worn application, rotate fungicide groups, use multiple modes of action, the right timing, right rate, and appropriate water volumes. So when we look at individual diseases, I just want to focus on anthracnose and lentils because that was a hot topic this summer, and that's where sort of the newest levels of insensitivity have developed. When we look at the charts in the seed guide, we see that there is some resistance to anthracnose race 1. We see some good versus fair in terms of variety resistance. However, what we're finding is that with anthracnose, there's actually 2 races. There's race 1 and race 0, and race 0 is the most predominant race. And to date, and with the exception of maybe what Tom was talking about, or Lori was talking about here today with CDC Grim, so far there hasn't been any resistance to race 0. So, you know, that's promising that that new variety, CDC Grim, did show an improved anthracnose resistance. So we look forward to seeing if that variety can be kind of the next level of resistance moving forward. In 2021, when we're thinking of anthracnose and how to manage for anthracnose, considering there could be insensitivity in the fields, the biggest thing is scouting regularly and seeing those first early symptoms. Be proactive, use your risk assessment, so using the charts that are available to you to help you predict the risk in the field, and then understand what your fungicide choices are. So know the products and know how they can be used to manage insensitivity. If your fields are at high risk for insensitivity, and the 2 key things are, have you had severe anthracnose problems in the past, and have you used a lot of group 11 fungicides? Then you may want to look at our checklist. So we do have tables. This is just a snapshot of part of the table, and it in these tables it outlines the different fungicides, and if you suspect you have group 11 insensitivity in the anthracnose, this table will help you choose the fungicides that could be the most effective in your fields. So this is just one of the chunks of the table, and it's showing that Elatis is a product that has both strobe and a non-strobe component, but the non-strobe component also has activity on anthracnose. So there's a number of products that are out there that have dual modes of action, but not both modes of action actually have activity on anthracnose. So knowing what products have activity is going to be important, and you can get that table out of our fact sheet. The last thing I want to talk about quickly is root rots and reducing risk in peas and lentils for going into 2021. So the first thing is testing the soil. There's bait tests and DNA tests that are available, and there's labs that offer the different services. We also have a fact sheet and resource that outlines that in more detail. The next thing is looking at your field history and understanding how many times you've grown peas or lentils in that same field over the past 20, 30 years. A lot of our fields in Saskatchewan are getting where we've had at least peas or lentils grown more than 5 times on that field. And that's typically when you can start seeing the buildup of some soil residual or soil pathogens. understand what the symptoms were the last time that you grew peas and lentils in this same field. Was there hot spots? Was the whole field affected? Was maybe there's some lodging, which is a good indication of late season infection? And also really looking at the environment that the last time peas and lentils were grown. And this is a hard one because it's hard to go back and really think about what happened. So what we did was we looked at the Environment Canada drought maps and developed this table that is available in the fact sheet as well. And it outlines for the different areas of the province by the different years what the general rainfall was. Was it average? Was it below average? Was it above average? Sort of highlighting the years where if peas or lentils were grown, they would be at higher risk for root rots. And so, for example, this map here is from 2017, and I picked 2017 because a lot of growers are on a 4-year rotation, so 2021 would be going into fields that could possibly have had peas or lentils 4 years ago. Now, if you're on a 2-year rotation, you want to look at 2019, 3 years, so forth. So if you look back at 2017, the Northwest area was the only area in the province that actually had above to or average to above average moisture. The rest of the province was below or below to average, which suggests, you know, maybe slightly lower risk compared to if you were growing peas or lentils on a field where peas and lentils were grown in 2016. We know that there was a lot of root rot issues this past year, and a lot of those fields were fields that had peas or lentils in 2016. So hopefully this chart can help you look back over your records and see when peas or lentils were last grown and what the moisture might have been to help you predict what your risk is going into your fields this year and what fields might be most suited. The other things to consider with root rots and reducing risk is rotations. So considering other pulse crop options in your rotation, there's faba beans, chickpeas, which have a level of resistance. Soybeans and fenugreek are non-host crops. Alfalfa and dry beans, depending on the varieties, have certain levels of resistance. The other thing is to look at crop impacts on oospore loads. So different crops have been suggested to potentially reduce spore loads over time or cause the spores, the oospores, to germinate in the presence of a non-host, so hopefully drop the levels. Things like mustard, oats, fall rye are things that are being evaluated in research that may help you with your root rot levels or your levels of Aphanomyces within the soil. The other thing is field characteristics. So looking at texture and drainage, managing water, you know, is a big thing. And as Tom talked about, it's not just managing Aphanomyces, it's managing Fusarium as well. So you might want to avoid field cereal residue that has really heavy Fusarium head blight. Even though it's not Fusarium graminearum, there are other Fusarium species that show up in Fusarium head blight that can also lead to Fusarium root rots. So avoiding those, but also looking at levels of resistance. As Tom talked about, there is can be slight differences with different varieties in terms of their tolerance to Fusarium. And we do have these tables in another resource on the root rots. So this is an example with peas and showing the different types of peas and the different varieties and the level of resistance that was found. And this was work that was done by the Crop Development Center with Dr. Sabine Benitza, who used the regional variety trial varieties last year or 2 years ago. It was done in 2019. And tested them with Fusarium avineaceum to see what their levels of resistance were. And what they really found was that the peas with the tannins in the seed coat did have a level of much lower level of disease than the non-tannin ones. So we know tannins in the seed coat can help with some of those seed-borne diseases. And that there was slight differences with some of the varieties, and I look forward to seeing more work on the Fusarium side. It's one of the components of the root rot complex, and it's about managing both the Fusarium and Aphanomyces, because when the two together, that's when the real problems arise. And then the last three things is looking at seed quality and insect pressure. We know that pea leaf weevil in combination with Fusarium makes things worse. We know nutrient management is also something to consider. If anytime you have roots that are not healthy, that means that they aren't able to function properly and really do that normal scouring and looking into the soil to access those nutrients that are throughout the soil profile. So making sure that there's enough nutrients that are available close proximity to those roots may help with protecting some of that yield. And then weed management is sort of the last thing, because it's not only crops that are susceptible to some of these pathogens, weeds can be as well. So things like shepherd's purse and chickweed are two examples of weeds that are also susceptible to Aphanomyces and can act as hosts. So just to summarize the root rot thing, we do have a reducing the risk of root rots fact sheet, and it has a nice checklist that will help you walk through to help identify the risk that you have in your fields, and a number of resources that are available on our website. So in summary, covered, seed quality. It's very important at this time of year, know our 1,000 kernel weights and disease levels. The chickpea health issue, variety isn't as big a concern anymore. It's about kind of reducing those stressors and keeping the plants as healthy as we can going into this year. Fungicide insensitivity, is variety choice really an option? In some of the crops it is. Some crops have definitely different tolerances for, say, Ascochyta. We know lentils, for example, have really good tolerance for Ascochyta, but lack that tolerance for anthracnose, except for possibly the new CDC Grim. And then choosing the right fungicides. Anthracnose, as I said, variety choice won't help, with the exception of possibly that new variety. And then root rots, variety choice may influence the fusarium side, but is not going to influence the Aphanomyces that we know of. And it's knowing, the two most important things are knowing what the soil and conditions the last time the crops were growing. That's the most critical. And with that, I'll open it up to any questions. And just a sneak peek that our Premier Pulse virtual series, so that's the new regional meetings, will be held starting February 2nd. And these are virtual events with the different crops being listed for the different dates. And it will consist of 2 hours of a live webinar followed by hot topics, including hot topics by crop, live presentations. There will be an agronomist panel, and the registrations are open. So with that, thank you. And I will entertain any questions if there's time.
We have no questions for you, Sherrilyn.
Thank you so much, Sherrilyn. We really appreciate you sharing your expertise with us. You always bring a lot of insight into these important topics. So we really appreciate you taking the time to talk to us today.
Thank you.